Welcome back to the deep dive. Today, we are tackling a subject that, honestly, it feels like it was ripped straight out of a Philip K. Dick novel. Or, maybe a Cronenberg film, depending on your stomach for biology.
Oh, definitely.
But I assure you, based on the stack of research we have on the desk, this is very much our reality. This is late 2025, early 2026 stuff. We're talking about mini brains.
It is a term that really grabs you, doesn't it? It immediately conjures up this mental image of a tiny, fully formed brain just floating in a jar of green liquid.
Yeah, maybe with little lightning bolts coming off it.
Right. Maybe it's plotting world domination or, telepathically controlling the lab assistants.
Exactly. It's the classic sci-fi trope. I think everyone pictures that scene from Futurama or those old B movies. But the reality, as we are going to find out, is it's significantly more complex and honestly maybe even more interesting than the fiction.
I'd say so.
The scientific term, and we should probably stick to this to be accurate, is human brain organoids, or HBOs.
Right. HBOs. And we are currently in the midst of what many researchers are calling a revolution. A genuine revolution in neuroscience and regenerative medicine. The sources we're looking at today are heavy hitters. I mean, we have consensus papers from the Asia-Pacific Neuroethics Working Group, deep technical reports from Parse Biosciences, and major studies published in journals like Frontiers and JMB.
So our mission today is to unpack this revolution. We really need to understand how a stack of stem cells in a petri dish is somehow replacing animal testing, powering what they're calling biological computers, and forcing us to completely rewrite our ethical rulebooks.
It's a big remit.
Yeah.
But at its core, this is a story about how we have finally found a way to model the human brain, but outside of the human body.
Okay, so let's start with the basics then. What exactly is an HBO? Because I think people hear organoid, and they might just picture a generic blob of tissue, like a meatball in a dish.
Well, it's a bit more than a biological meatball, but it does start humbly. An HBO is a three-dimensional biological structure grown in vitro, so in the lab, from human stem cells.
But the key characteristic here, the thing that makes it so special and separates it from just a clump of cells, is that these cells self-organize.
Self-organized. That's the wild part to me. You don't have a microscopic architect in there telling them where to go.
It is wild. And they aren't just flat layers of cells sitting in a disk, which is how we used to do it in old school biology. They develop architecture. They form distinct regions that mimic the developing human brain. We're talking cortex, ventricles.
So they're not just a random collection of cells. They're actually building their own neighborhoods, essentially.
That's a perfect way to describe it. They know who belongs where. They develop cellular diversity, producing neurons, astrocytes, and microglia. They mimic the cytoarchitecture, the cellular arrangement of the fetal brain.
Incredible.
Now, to understand how we get there, we have to look at the recipe. How do you actually grow a brain in a lab? Because it sounds like magic, but it's actually a very specific set of instructions. Right, let's get into the kitchen. Segment 1, the recipe. I was reading through the Parse Biosciences reports, and it sounds like alchemy, but it's actually really rigorous science. It all starts with pluripotent stem cells.
Yes. Specifically, induced pluripotent stem cells, or iPSCs. This was the Nobel Prize-winning breakthrough a while back, but it's the foundation of everything we're discussing today.
Okay, so break that down for us. What does induced mean here?
It means you can take a donor skin cell or a blood cell, a fully mature adult cell, and you reprogram it. You force it back to a blank slate.
It's like hitting the factory reset button on a cell, or using a time machine to send it back to when it was an embryo.
That is a perfect way to visualize it.
Yeah.
You use what are called transcription factors, specifically a cocktail of proteins, OCT4, SOX2, KLF4, CMYC. These proteins essentially go into the cell's DNA and wipe its memory. You are no longer a skin cell. You are now a stem cell.
And now it's pluripotent. It can become anything.
Anything. So, to make a brain organoid, you first get these cells to clump together. They form what are called embryoid bodies. Then, you use a process called neural induction to push them toward becoming neuroectoderm. That's the tissue that eventually becomes the entire nervous system.
So you nudge them, you give them a little push, like, hey, don't become a liver, become a brain. And this is where that matrigel comes in, right? I saw that mentioned a few times in the methodology sections.
A crucial ingredient. Matrigel is a protein scaffolding. Think of it like a jelly or a gelatin. It mimics the extracellular matrix, the goo that holds our cells together in the body.
So it gives them something to hold on to.
Exactly. You embed these embryoid bodies in matrigel droplets. This gives them a 3D structure to latch onto. If you didn't do this, they just collapse into a puddle. Then, To make sure they grow properly, you put them in a bioreactor.
Which is essentially a spinning flask. I've seen pictures. It looks like a high-tech blender set to very slow.
It is a spinning bioreactor, yes. It just agitates the fluid. And this isn't just to mix it up. It improves nutrient absorption and oxygen diffusion. Because these things don't have hearts or pumps, they need that movement to breathe, so to speak.
So the movement forces the nutrient-rich liquid into the crevices of the tissue.
Precisely.
Now, looking at the literature, there seem to be two main philosophies on how to let these things grow. You have the unguided approach and the guided approach. It sounds like parenting styles.
So that's actually a fantastic analogy. It really is. The unguided or intrinsic method is like free-range parenting. You provide the environment, the matrigel, the bioreactor, and then you just let the cells do what they want. You trust the internal genetic programming.
And what do they want to do?
They want to become brain tissue. The advantage here is that you get this high diversity of brain regions in one clump. You might get some cortex, some midbrain, some retina, all mixed together in a sort of chaotic mosaic.
Is there a downside to that free-range chaos? I imagine there has to be.
Absolutely. It's inconsistent. One batch might look totally different from the next.
Right.
Which is a nightmare for a scientist trying to do a controlled experiment.
Of course.
If your control group looks different than your test group just by random chance, your data is useless. You can't publish that.
Right. So that leads us to the guided or regionalized approach, the other parenting style.
This is the helicopter parent method. You use chemical inhibitors and activators, specific molecules, to force the tissue to become a specific part. You will be a cerebral cortex. You will be a cerebellum. It's much more controlled and reproducible. You know exactly what you're getting in every jar.
But even with that guided method, there's been a historical problem, right? I read about the necrotic core. It sounds like a heavy metal band, but it's actually a serious biological limitation.
It's a major limitation that held the field back for years. As the organoid grows, the outside gets plenty of oxygen and nutrients from the fluid in the bioreactor.
Yeah.
But the inside, it's too far away. The tissue gets too dense. Without blood vessels, the nutrients can't diffuse deep enough. So the center starves and dies. That's necrosis.
So you end up with a living shell and a dead, rotting center. That's horrifying, but also practically useless if you want to study deep brain structures. How are they fixing that now in 2025?
Vascularization. They're building plumbing. There are a few ways to do it. Some researchers are incorporating endothelial cells, those are the cells that line blood vessels, right into the initial mix.
So you just sprinkle them in with the stem cells?
Pretty much. They might use HUVECs, or they can genetically engineer the cells to express a gene called ETV2, which induces blood vessel formation.
So the organoid grows its own veins.
Exactly. It vascularizes itself.
Yeah.
And I saw something about organ-on-a-chip technology, too, in your notes.
Yes, microfluidics. This is really cool. Instead of just spinning them in a jar, you grow the organoid on a silicone chip with tiny microscopic channels that actually pump fluid through the tissue.
So it mimics blood flow.
Exactly. It reduces stress on the cells and keeps that core alive, which lets the organoids grow much larger and more complex. It's like installing an artificial heart and circulatory system for this little piece of tissue.
In which you have these larger, healthier pieces, you can start doing something really cool. Building assembloids.
I love this term, assembloids. It sounds like a Lego set for biologists.
It's exactly what it sounds like. You take two different regionalized organoids, say a cortex and a striatum, and you fuse them together. You physically place them next to each other.
Why would you do that? Just to see if they stick?
To watch the connections form. In the human brain, these regions talk to each other. We want to see how they establish that line of communication. You can watch neurons migrate from one region to another. You can watch axons, the long cables that neurons use to talk project from the cortex into the striatum.
So you're literally watching the brain wire itself up in real time?
In a dish. It's unbelievable.
That is incredible. And according to the reports from late 2025, we've reached the level of multi-region brain organoids, or MRBOs.
That was a massive leap. They used a sort of biological superglue to integrate cortex, midbrain, and hindbrain all into one system. These MRBOs are replicating about 80% of the cell types you'd find in a 40-day-old fetal brain.
Wow.
That is a level of complexity we couldn't even dream of 5 years ago. We aren't just looking at pieces anymore. We are looking at systems.
Okay, so we can build them. We have the recipe, we have the plumbing, we have the superglue. But why does this matter to anyone outside of a lab?
This brings us to the regulatory shift. because something huge happened in the spring of 2025 involving the FDA that changes the game for medicine.
This cannot be overstated. The FDA announced plans to phase out mandatory animal testing for monoclonal antibodies and a lot of other drugs.
Right.
For decades, if you wanted to get a drug approved, you had to test it on animals. It was the gold standard. You did mice, then maybe rats, then maybe primates.
But it wasn't a perfect standard, was it? We've all heard stories of things working in mice and then just failing spectacularly in humans. Far from perfect. We call it the lost in translation problem. A mouse brain is not a human brain. They have different gene expression. They lack the complex cortical solding we have. Our brains are wrinkly, there's a smooth.
Okay.
And very often, a drug would work beautifully in a mouse to cure a mouse version of Alzheimer's and then fail miserably or even act toxically in a human trial.
That's billions of dollars and years of time wasted on false positives.
Exactly. So the organoids are the solution to that translation error. They are human tissue with human DNA and human architecture.
They are the new standard. The NIH even established a new office in April 2025, ORIVA, the Office of Research Innovation, Validation, and Application, specifically to standardize these organoids.
Which is so important.
Because if you're going to replace the lab rat, you need to be sure the replacement is consistent. You can't have artisan organoids if you're testing safety.
And speaking of consistency, I have to mention the Stanford study. This is one of those details that sounds mundane but is actually revolutionary. They solved the clumping problem using food additives.
This is my favorite weird fact. Tell us about the food additives.
It's amazing, isn't it? One of the issues with mass producing these things was that they would stick together in the bioreactor. If they clump, they grow unevenly, and your experiment is ruined.
Right.
Stanford researchers found a common food additive, something you find in a grocery store, that prevents that sticking. It coats them. And it allowed them to grow thousands of identical mini brains at once.
That is scaling up. That turns a boutique experiment into an industrial process.
It is industrial scale. And the proof is in the pudding. They screened 298 FDA-approved drugs on these mass-produced organoids. These are drugs already on the market, drugs people take every day. And guess what?
What did they find?
They found toxicities, specifically stunted brain growth, that the animal models had completely missed.
That is a little terrifying, considering those drugs were already approved and are in pharmacies right now.
It is, but it validates the shift. The human model found the human problem. It shows us what we've been missing by relying on rodents for so long.
So that leads us directly into what you might call the killer app for organoids: disease modeling and precision medicine. This is where it gets personal. This isn't just about general research. It's about your specific biology.
It does. We are moving toward the concept of the digital twin, or maybe more accurately, a biological twin. Imagine taking your cells, growing a model of your brain or your tumor, and testing drugs on that instead of on you.
The cancer applications for this seem particularly advanced. I was reading about this QPOP platform for glioblastoma. Glioblastoma is a scary one, right? The really aggressive brain cancer.
Glioblastoma is an incredibly aggressive brain cancer. The prognosis is usually very poor. One reason it's so hard to treat is that every patient's tumor is slightly different genetically. What kills my tumor might essentially be vitamin water for yours.
So currently, doctors have to kind of guess. They follow a protocol, but it's not personalized.
They follow a standard of care, but it's often a bit of a gamble. So the QPOP platform takes the patient's actual tumor cells during surgery, grows what they call tumor avatars, patient-derived organoids, and then blasts them with a whole panel of different drugs.
So you're shotgunning treatments at the avatar to see what sticks. You're sacrificing the clones to save the original.
Exactly. But it's smarter than just shotgunning. It's the quadratic phenotypic optimization platform. An algorithm analyzes the results and identifies the best drug combination for that specific patient in real time.
Wow.
You aren't guessing. You know what kills the tumor because you just watched it happen in the dish.
That is a game changer. It completely removes the trial and error phase from the patient's body. But it's not just cancer. The Alzheimer's research coming out of this is fascinating, specifically regarding the APOE4 gene.
Right. We've known for a long time that the APOE4 gene is a major risk factor for Alzheimer's. If you have it, your odds go up significantly. But it was hard to pinpoint exactly why in a living brain.
Right.
Using the MyBrain platform, which integrates neurons, glial cells, and blood vessels, researchers made a breakthrough.
They isolated the glial cells, right? Those are the support cells, the so-called glue of the brain.
Yes. For a long time, we thought neurons were the main actors in Alzheimer's. But they proved that APOE4 glial cells alone even if the neurons were perfectly healthy and normal, could drive the accumulation of amyloid beta plaques.
Wait, so the support staff is causing the problem, the janitors are messing up the whole building.
Exactly. That's a huge discovery. It means we have a specific cellular target, the glia for therapy. We wouldn't have seen that clearly in a mouse model because mouse glia behave differently.
So we might have spent 20 years trying to fix the neurons when the problem was the glia all along.
It's very possible.
And then there's infectious disease. We learned a lot about this during the post-2020 era. Organoids helped us understand how viruses attack the brain.
Absolutely. Take HIV. For years, we wondered why patients on antiretrovirals could still have cognitive issues. Organoids revealed how the virus hides in reservoirs within microglia, the brain's immune cells. It showed us how the virus causes neuroinflammation even when viral loads are low.
And with COVID-19, I remember all the headlines about brain fog. People were really scared by that.
And for good reason. Studies on organoids show that SARS-CoV-2 specifically infects the choroid plexus.
That's the barrier defense of the brain, the main wall.
Correct, it's the gatekeeper. And the virus was neurotoxic to tau proteins, which are implicated in Alzheimer's. This provided a biological explanation for the brain fog and neurological symptoms so many people experienced. It wasn't psychosomatic, it wasn't just stress.
It was a direct viral attack on specific brain structures, and we could see it happening in the organoids.
We could see it. And we can't forget Zika. That feels like it was the turning point for this technology, the moment it really went mainstream.
It was.
Organoids were instrumental in proving the link between the Zika virus and microcephaly, the stunted brain development in fetuses. Before that, it was just a correlation. But with organoids, they could see the virus attacking the neural progenitor cells, the baby brain cells, in real time. It ended the debate.
It's incredibly powerful. But there's another application that sounds a bit more violent. Traumatic brain injury, or TBI. They are using blast simulators on these things.
Science isn't always gentle. To understand concussions, blast injuries from war zones, or sports injuries, researchers are using benchtop blast simulators or compression devices to physically impact the organoids.
They're giving the mini brains a concussion.
Effectively, yes. They hit them with shock waves, and then they can observe neurodegeneration, the breakdown of the connections, and cell deaths in real time under a microscope. It's a way to study TBI without hurting a person or an animal and seeing exactly what happens at the cellular level the moment impact occurs.
You can see if the cell bursts, if the connections snap.
All of it. Now we can look.
Okay, so we've got disease modeling, we've got drug testing, but now we need to pivot to the part of the research that feels the most futuristic. Segment 4. Organoid intelligence, or OI.
The computer made of meat, as some have crudely called it.
It's a vivid image, but the logic behind it is actually pretty sound. We are hitting a wall with silicon computing, aren't we? Especially with energy.
We are hitting an energy wall. Think about AI. Data centers running modern AI models consume megawatts of power. They require their own power plants. They generate massive heat. But the human brain, it runs at about 20 watts.
It's less than a dim light bulb. It's absurdly efficient.
But yet it stores exabytes of data and processes information in ways silicon still can't match, especially when it comes to learning from very little data. So the idea behind biological computing is to use the natural computational power of living neural networks. Why build a fake brain out of silicon when we can grow a real processor?
How does that even work? You don't just plug a USB into a blob of tissue?
Not exactly. You use what's called reservoir computing. You rely on the organoid's natural synaptic plasticity and its chaotic internal dynamics to process data. You feed information in as electrical signals, the organoid reacts, the ripples propagate through the tissue, and you read the output. You don't program it, you train it.
And they've actually done this. This isn't theoretical anymore.
No, the benchmarks in 2025 were impressive. There was a system called BrainAware that achieved 78% accuracy in speech recognition. It learned to identify voices from audio files that were converted into electrical pulses.
That is wild. A blob of cells identifying who is speaking.
And there was a Braille recognition study. A 3-organoid ensemble achieved 83% accuracy in classifying Braille letters. They fed the spatial pattern of the Braille bumps as electrical signals, and the organoids learned to distinguish A from B.
And of course, we have to mention Dishbrain. This was the story that broke the internet.
The Pong player.
The Pong player, tell us about it.
In vitro, neurons learn to play a simulated game of Pong. The researchers hooked the neurons up to the simulation. If the neurons fired in a way that moved the paddle to hit the ball, they got a predictable electrical signal back. If they missed, they got a chaotic, random signal.
And neurons hate chaos. They crave predictability.
Exactly. It's called the free energy principle. Neurons want to minimize surprise. So they learned to play Pong, not because they like video games, but because playing well made their world predictable and comfortable.
And they exhibited what the researchers strictly defined as sentience, which is a loaded word.
A very loaded word.
Yeah.
But they defined it strictly as responsiveness to stimuli.
So how do we interface with them? What is the actual hardware here?
It's a hybrid system. You use CMOS microelectrode arrays, or MEAs. These are chips with thousands of tiny electrodes that the organoid sits on. It's like a bed of nails, but microscopic and conductive. The MEAs read the electrical firing of the neurons and can also send electrical signals into the organoid. It's a two-way street.
There was one finding in the research that really stopped me in my tracks. They called it the pre-configured operating system. Can you explain that?
That was a profound discovery, I think, from UC Santa Cruz. They looked at organoids that had never seen or heard anything, no sensory input whatsoever. And they found that these organoids develop default mode electrical patterns anyway.
So before they see or hear anything, they already have a pattern. They're humming a tune to themselves.
Exactly. It suggests that the brain has an innate blueprint, a pre-configured operating system for processing information. We aren't born as blank slates. The hardware comes with software pre-installed, ready to organize the data it's about to receive.
That changes how we think about human development entirely.
It does. It's philosophically heavy.
Which is the perfect segue to our final major segment, the
Yeah.
ethical minefield. Because you can't talk about growing human brain tissue and teaching it to play Pong without asking some very hard questions.
And this is where the Asia-Pacific Neuroethics Working Group consensus paper is so valuable. We have to separate the signal from the noise here.
The noise being the red herrings, the sci-fi stuff.
Right. The media loves to portray these as fully conscious mini-brains, trapped in jars, screaming silently. That is the hype. It sells clicks.
And the reality.
The reality is that currently, these are structurally simplified models. They are roughly equivalent to parts of an early prenatal brain. They lack sensory input. They lack a peripheral nervous system. They aren't thinking in the way we understand it.
And focusing on that sci-fi scenario distracts from the real immediate risks.
Exactly. Like consent. This is a legal nightmare waiting to happen.
It seems like it.
Let's say you donated skin cells for research 10 years ago. You signed a broad consent form. Did you agree to have your genetic material turned into a biological computer?
Probably not. I thought you were just looking at my cells under a microscope to study, I don't know, aging.
Did you agree to have it commercialized as part of a neural network that runs a smart home or a weapons system? And here is a turn-on one. Withdrawal. In research, you can usually withdraw your participation. You can say, I'm out, destroy my samples. But can you withdraw your organoid if it has become an integral part of a biological computer? Or if it's been fused into an assembloid? If my cells are the cortex and your cells are the striatum and I want out, do we have to kill the whole thing?
You can't just unbake the cake.
Precisely. And what about incidental findings? What if the organoid reveals that you, the donor, have a genetic predisposition for a terrible neurodegenerative disease that you didn't know about? Do the researchers have a duty to tell you? Do you even want to know?
Or is that a massive invasion of your privacy? A privacy nightmare.
I know.
But even if we dismiss the brain in a jar fear for now, the consciousness question is getting more nuanced, isn't it? We aren't just asking, is it conscious anymore?
No, we have to define our terms. There's a huge difference between sentience, the capacity for valenced experience like feeling pain or pleasure, and higher order self-awareness. You know, a lobster might feel pain, but it doesn't write poetry about it.
Right. And China actually set some strict boundaries on this in May 2025. They drew some lines in the sand.
They did, the red lines. They prohibited implanting human brain organoids into animal reproductive systems, which prevents some very dark sci-fi scenarios. But more importantly, they mandated monitoring. If the electrophysiological complexity of an organoid approaches certain thresholds of consciousness, the research has to stop.
But how do you test for that? How do you know when the light comes on inside the tissue?
That is the million-dollar question. We need a Turing test for organoids. We are developing new tests to assess whether these biological computers are developing inner lives or problem-solving capabilities that imply they have moral status. We are looking for perturbation responses. If we poke it, does it react in a complex, integrated way?
And then there are the chimeras. We touched on this with the rats. This feels like the island of Dr. Moreau.
Yes. Transplanting human brain organoids into rat brains. We know they integrate, We know they vascularize, and recent studies showed they can even respond to visual stimuli. Flashing lights in front of the rat's eyes triggered a response in the human tissue.
So the rat sees through the human brain tissue.
Effectively, the human tissue is processing the visual signal. The rat's eye sends the signal, and the human organoid interprets it. This creates what ethicists call moral confusion. Does a rat with human brain tissue deserve human rights?
That sounds absurd on its face, but if it has a piece of human cognition, it gets blurry.
The consensus right now is no, but it requires strict justification. You can't just do it for fun. It has to be necessary for medical progress and proportional. But the line is getting blurrier every day.
It feels like we are walking a very, very tightrope. On one side, you have the potential to cure Alzheimer's, to revolutionize computing, to save millions of lives. And on the other, creating something we don't fully understand and might not be able to ethically control.
That is the nature of paradigm shifts. We have moved from 2D cultures to 3D organoids to assembloids, and now to integrated biological systems. We are bridging the gap between clinical history, looking at the past, and personalized prediction shaping the future.
It's a reminder that these aren't just tools. They are derived from us. They are us, in a very real biological sense.
They are. And that is why the human element is so important. We are doing this to help human patients, to stop the suffering caused by diseases we've failed to cure for centuries. We have to keep our eyes on that prize while we carefully navigate the ethical minefield.
So as we wrap up this deep dive, we always like to leave you with a provocative thought. Something to chew on.
Here's mine. We are racing toward organoid intelligence that rivals silicon efficiency. We are planning to integrate these biological processors into machines. At what point does the computer become a patient with rights? And maybe more personally, if an organoid grown from your cells develops a memory, is it your memory?
A terrifying and fascinating thought. Thank you for joining us on this deep dive into the revolution of human brain organoids. We will see you next time.